Well, welcome everybody to our discussion today on the future of immunotherapy for patients with gastrointestinal stromal tumor. I'm delighted to be joined on this panel with several key individuals in this field. Dr. Junaid Arshad is a medical oncologist who sees GIST patients at the University of Arizona. Ron DiMatteo is a surgical oncologist who sees patients at Penn and has a research lab investigating new ways to manipulate the immune system against gastrointestinal stromal tumor cells. And Arun Singh, who's a medical oncologist at UCLA Johnson Comprehensive Cancer Center, who has been active in clinical trials utilizing immunotherapy for patients with GIST. I'll begin by introducing Junaid Arshad, who's going to give us a bit of an overview in immunotherapy and the immune system as it pertains to gastrointestinal stromal tumor. And then we'll hear from Ron DiMatteo and Arun Singh, and then we'll have questions at the end, wrapping up around 2:15. Without further ado, go ahead, Junaid.
Thank you very much, Dr. Trent, for the introduction, and good afternoon, everyone. I would like to take a moment to just say this word here. This has been it's my pleasure to give a talk about overview of immunotherapy with some of the best known people who've been treating the gastrointestinal stromal tumors. And I would especially like to thank Dr. Jonathan Trent, who is my mentor. And I owe him all the knowledge he has taught me so far. Thank you very much, Dr. Trent. I will share my screen, and I have only a few slides. I try to keep it simple for everyone to. Okay. So we will be starting with a few introductory slides, and we'll go into the concept of immunotherapy and immune system and some of the important things to understand before we discuss the immunotherapy targets and the clinical trials. I have no disclosures. So just introduction about gastrointestinal stromal tumors, commonly referred as GIST. Are a subset of sarcomas which arise within the gastrointestinal system and spread within the abdomen. Relatively rare group of neoplasms, 0.2% of all GI tumors and 80% of sarcomas. Peak incidence is peak age is between 40 to 60 years of age, and men and women are affected equally. Majority of the patients have metastatic disease at the time of presentation. The prevalence of the disease is higher than the incidence because the clinical course can last almost up to 10 to 15 years. The genetic mutations are very important because they drive the phenotype and ultimately the treatment landscape of the GIST. This slide kind of shows that surgery remains the curative treatment, and surgery remains the mainstay of the treatment. It's curative in 40 to 60% of the cases. Patients with locally advanced or metastatic GIST are notoriously resistant to chemotherapy, and the treatment of these locally advanced and metastatic GIST is based on the presence of driver mutations. Approximately 75 to 80% of GIST tumors have KIT mutations, and the patients who do not have KIT mutations are considered wild type for these mutations and have other mutations like PDGFR, RAF, NF1, NTRK, and some of the other mutations. So currently there are 5 medications that are FDA approved in the setting of metastatic GIST treatment. These include imatinib, sunitinib, regorafenib, and repotrectinib in the first, second, third, and fourth line settings. Avapritinib is a specific target for D842V mutant metastatic or locally advanced GIST tumors. So why do we need, so we have all these wonderful medications that we are using in the treatment of GIST. Why do we need more treatments? None of these treatments are curative. All patients who are on these treatments at some point definitely will progress with their disease, and we always need a new treatment target. So this represents an area of huge unmet need for new therapy targets, for newer combination treatments, and even for treatments which have better side effect profile. So talking about immunotherapy, what is immunotherapy? Immunotherapy is a treatment of the disease, in this case, like cancer, which acts by either activating or suppressing the immune system. There are like 4 big domains of immunotherapy. It can be either antibodies, which are like proteins that bind to cancer cells and tag them and kind of be identified by the immune cells for them to be cleared. There are checkpoint inhibitor medications, the medications which bind to the checkpoint proteins and kind of gives a immune system and prevent those inhibitory signals which are preventing the immune system from clearing those cancer cells. There can be vaccines, which are foreign materials, which can be introduced into the body to train our immune system to kill the cancer cells. And then there are like some T cell transfers that we remove the T cells and modify them and then reintroduce them into the body so that they can identify the cancer cells for immune clearance. So these were just like the basic principles of the type of immunotherapies. In order to understand the concept of immunotherapy, we have to understand the immune system. And it is a very complicated process and a lot is involved in this. I'll try to make it simple, and for that it's for everyone to understand. So our immune system has 2 arms, an innate arm and an adaptive arm. So an innate arm is the first line of defense. It is non-specific, it is rapid. It does not require a prior exposure to any infectious agent. And it includes certain cells like dendritic cells, macrophages, and neutrophils. And there are certain biologically active amines, which are called cytokines, which are biochemicals which are used by these cells for crosstalking. So the important ones are like alpha interferon, which causes gamma interferon, and then that acts on natural killer cells and some of the other cells. So similarly, like adaptive immune system is the second line of defense. It requires a prior exposure to an antigen or to an infectious agent. It takes days and weeks to mount. It consists of T cells and B cells. Now there are different types of T cells. So there are CD8 cytotoxic T cells. I usually, for understanding purposes, will call them as the military cells. These are the CD8 cytotoxic military cells. And there are helper cells like T1 helper cells or T2 helper cells, which kind of help regulate the CD8 cytotoxic cells. And then there are regular Treg cells, which are involved in chronic inflammatory responses, and they're also involved in causing immune suppression in cancers and serves as a mechanism of escaping the immune system. What really happens is like these 2 systems work together, and there is a balance between stimulatory and inhibitory signals to maintain an effective homeostasis environment. So just relative to our topic of GIST, some of the components of the immune system that will be relevant, like alpha interferon, which causes an increase in gamma interferon, and gamma interferon stimulates natural killer cells and the CD8 cytotoxic cells, which are the military cells. And then certain biologic amines, like I mentioned, indole diamine oxygenase, and a short form is IDO, it suppresses natural killer cells and these military cells to maintain an effective balance. So just to remember that there are positive signals for these immune cells and there are negative signals for these immune cells. So what happens in the GIST microenvironment? So what happens in the GIST microenvironment? What kind of cells are involved? So GIST microenvironment has like certain macrophages, which are called like tumor-associated macrophages type 1, tumor-associated macrophages type 2, and these cells, and in addition to these cells, there are some other cells like CD3 type of T cells and natural killer cells and gamma delta cells, T cells and B cells. So what cells are important in this? So type 2 macrophages are one of the most important cells that are present in the GIST microenvironment. And these type 2 macrophages cells are related to kind of determine the number of Treg cells, or their T regulatory cells. Just to remind, T regulatory cells are the cells that were involved in causing the immune escape for these cancer cells. Now, higher the number of Treg cells, it causes a decreased ratio of CD8 cells, and it decreases the MHC expression. And MHC expression is like a protein which the T cells use in order to express the cancer cell antigens for them to be recognized by the immune system. There are some other cells too, like tumor infiltrating cells like CD3 plus cells and FOXP3, which is another type of protein that Treg cells express, and some of the other. This is a very complicated process, and with the ongoing work, there's better understanding of this microenvironment. So just to summarize what we learned in this slide is, because the GIST microenvironment has a lot of Treg cells, which causes immune suppression and causes a decreased activity of military cells. So in other words, just to summarize all this, a GIST microenvironment is immunosuppressive, meaning even if you have immune cells trying to attack the tumor within the microenvironment, the immune cells are suppressed. They're not able to carry out their job correctly or efficiently. So I would like to talk about a little bit of the immune signatures too, like how do we know, in other words, just to put a then an easy description of this is immune signatures are like different type of signatures which are actually used to determine like the type of these immune cells that are present in tumor samples. Now, some of these, the two commonly that have been recently, like in the published data that came forward, are the gamma interferon expanded immune signatures and T cell inflamed signatures too. So these immune signatures are an effective way of looking at, in other words, to determine what tumor types are more likely to respond to some of the immune checkpoint inhibitors, or like our immune medications. Now, some of these immune markers are not widely used and still are under study. But this is an effective way of looking at the microenvironment of the GIST tumors. So it has been studied that KIT PDGFR mutant tumors have a better immune signature profile as compared to the wild type tumors. And out of all these tumors, PDGFR and D842V mutant GIST tumors have the best immune signature profiles. And again, I said it kind of relates to the presence of CD8 cells, which are military cells, and M2 macrophages. What does imatinib does? How does imatinib play in this role of, they will be learning about from Dr. Singh and Dr. DiMatteo after this, that there are some combination clinical trials as well with tyrosine kinase inhibitors. So I just wanted to explain the effect of imatinib too. So imatinib, in other words, can play a very effective role in increasing the number of our military cells, CD8 cells. It can decrease or cause apoptosis of our regulatory cells, which are immune suppressive cells. It increases the chemical named gamma interferon, which causes increased activity of natural killer cells, and it suppresses the inhibitory cytokines like IDO, which inhibits the military cells. So in other words, imatinib effectively works to enhance the immune environment of GIST tumors, and sometimes these combinations can be used to remove the resistant clones as well, the tumors which are not responsive to imatinib. So I included this slide before I'm running towards the end of my time limit too. I just wanted to give like this basic slide to explain the effect of immunotherapy, action of immunotherapy, like how these things work. So if you look on the extreme left, that the T cell receptors have this antigen, which is a portion of the cancer cell, and they need this for the identification of the tumor cells. And then now this T cell gets inhibitory signals and the positive signals. So CD28 and CD80 over here is a positive signal that promotes the T cell to carry out its job. But if you look on the right, there are two negative signals too, like CTLA4 and B7 or CD80. The cell shown below is a tumor cell, and this T cell has this negative receptor on it, and this tumor expresses the CD80 marker, which kind of sends an inhibitory signal to turn off the T cell. And similarly is this PD-1 and PD-L1 interaction. The T cells have PD-1 receptors, and so these tumors start expressing excessive ligands which bind to these PD-1 receptors and kind of stops the T cells from causing the clearance of the cancer cells. And these are some of the medications you will be hearing just now in next few steps in the presentation that have been the important targets in clinical trials. This is my last slide. This is just like I copied it from one of the review articles I did with Dr. Trent earlier in the year. This slide was just to give you an overview of the immunotherapy targets. So basically it can be like cytokine-based therapies, like it can be like you can give more interferons, which can inhibit the Treg cells and can improve the natural killer cells. There was a phase 2 trial about this in stage 3 to 4 GIST tumors that did show some 1 complete response and 7 partial responses. There are definitely immune checkpoint inhibitors, which include pembrolizumab, some combination treatments with ipi/nivo. There is some of the data that has recently come out in CTOS. Dr. Trent shared about it, and I think Dr. Singh is present on one of those clinical trials as well. There are specific anti-KIT monoclonal antibodies, which are conjugated to microtubule mitensinoid. These are toxins which bind to the KIT receptors, and the whole drug conjugate is internalized, and they can work both for imatinib sensitive and resistant clones. There are some bispecific monoclonal antibodies and cell-based therapies as well. Just to summarize that the role of immunotherapy in soft tissue sarcomas is growing, and we will see future in some of the clinical trials that are coming forward for this. We are literally at this point that the role of immunotherapies in gastrointestinal stromal tumors cannot be ignored. Definitely there is one way to go, and that way to go is to go forward. Thank you very much. That was my last slide, and we will do questions in the end.
Great overview. You can stop sharing now. We'll transition to our next speaker, who's really established a number of important discoveries in his research laboratory. So I'm delighted to have Ron DiMatteo discussing his research in the interaction between the immune system and gastrointestinal stromal tumor. Thank you, Ron. I think you're muted.
It's sharing properly.
First, and then I'll go back. Okay, well, yeah, thanks for inviting me to speak. You know, it can be a little bit challenging because we have an audience of probably a variety of different understandings of the immune system. So I'm going to try to provide some general concepts, but also try to give a few specific examples. And I think the introduction, you know, was helpful. You know, most people, when they hear the immune system, they sort of run away because it's so complicated. And it's really one of the biggest advances in the last 5 to 10 years in clinical oncology. And so it's becoming more widely appreciated and understood, I would say. So I was going to talk a little bit about some of the immune aspects of GIST, but also sort of emphasize the technology to you that's going to allow us to do things that we could never do before, and to understand aspects of individual immune cells within the tumor, unlike any prior technology that we've had. So I have one small research grant from Blueprint Medicines. So, you know, this is sort of the overview of what we have available in the laboratory. So we have several mice that are genetically engineered to develop a GIST. And the original mouse was made by Peter Besmer at Sloan Kettering Institute. and we collaborated with him, and he eventually retired, and we're the primary users, I would say, of this mouse. So, you know, we have mice that have very similar mutations as to what occurs in human. Now, the only difference is there's one amino acid difference between mouse and human. So the 558 in the mouse is representative of a 557 KIT mutation in humans. And then you may or may not know that the 653, which is 654 in humans, is the most common way for a human GIST to become resistant to a tyrosine kinase inhibitor. And so we have a mouse that has its original mutation, but also this resistant mutation. And then, you know, these mice allow us to do all kinds of crosses. And I'm just listing a variety of immune deficient mice where they're lacking sort of one protein or one cell type. And that gives us incredible power to ask very specific questions. Obviously, everything at the end of the day has to be answered in a clinical trial. But as Dr. Singh will probably, you know, convey to you, it takes a while to do a clinical trial in humans, and we can answer questions in the laboratory, you know, much faster, and then test those specific outcomes to see if they're relevant to humans. We have a lot of human GIST specimens because I'm a surgeon, and you know, the patients are very willing to sign a consent form that allows us to study their tumor. We have all the conventional things you'd have in a laboratory: Western blot, immunoassays. We have this machine that basically can isolate individual cells from a tumor, or just analyze, you know, tens of thousands of cells in a very limited amount of time. And then the technology that I'm referring to is the following. And this is really a game changer for all of cancer and many other diseases, actually. So this is called RNA sequencing. And RNA sequencing allows us to figure out what's happening in an individual cell at the moment that we analyze it. So you may or may not know there's about 30,000 genes in humans and mice, okay? And so that's DNA. And each cell expresses only some of those genes, and they express them at different levels. And those levels change depending on the environment, the treatment, you know, all kinds of things. And so in the past, we could study like the entire tumor at once and say, okay, what genes are on, what genes are off. And then we'd have to kind of guess, you know, well, is that in the tumor cell? Is that in the immune cell? Is that in the cell that's lining the blood vessel? We don't have to guess anymore, because we can do this down to the level of any particular cell type within the tumor. We can tell you exactly what genes are being expressed at that moment. Now, there's some minor things about, you know, what genes are expressed, which means RNA. Those RNAs turn into proteins, and you know, it's not one-to-one. Not every RNA becomes a protein, but it's incredibly insightful to be able to do this. And I'm going to try to show you some of this. So here's our model that I mentioned to you in the mouse model. And this is a gross picture of the tumor. Unlike in humans, it actually happens in the first part of the small intestine. Why that is, you know, nobody's really answered that. It presumably is due to the precursor cells that exist in that part of the GI tract. Here's what they look like under the microscope, which is very similar to what we see in human GIST. And then this is actually a CT scan done in 3D, and these are like the leg bones, and this is the tumor that we've pushed into the pelvis just because it's easier to image. And these mice die at a defined rate if they're not treated. So this was published years ago. One of our first studies that we did in the laboratory was to do RNA analysis of the entire tumor, because that's all that existed at the time. So this was around 2009. We had 3 mice that were treated with sort of placebo, 3 mice treated with imatinib. You can't see it well, but there's 3 columns on each side here, so 6 total columns. And here we're analyzing basically all the genes that exist. And red means high expression, or red means, red means high expression of a gene. And then sort of bluish or gray means very low expression, like you can see here. This is the spectrum of expression at the top. And one of the first things that we found when we gave imatinib to these mice was one of the genes that changed the most was a gene called IDO. IDO had been known at the time for about 5-7 years to be important in cancer. And IDO has a very long name. It's an enzyme, it's a protein that helps sort of break down the amino acid tryptophan. And it turns out the products of the breakdown of tryptophan are very immunosuppressive, which is why tumors want to express IDO, because it sort of hides them from the immune system, or at least disables the T cells that are trying to kill the tumor. So this was kind of a striking finding, because everybody thought, well, imatinib only worked by inhibiting KIT signaling, but a byproduct of giving imatinib is that you reduce the amount of this protein called IDO, and that allows the immune system to attack the tumor a little bit better. Now, I call this the map of the world, because if you look and tilt your head, it sort of resembles the world. This is sort of North America, this is Central America. Well, South America doesn't look perfect. This could be Europe. Australia may be overrepresented. But this is actually the world of just immune response. So there are thousands of dots on this slide. In fact, there's about 50,000 dots. Each dot represents a cell, okay? So we took mouse tumors and we digested them after treatment or after placebo, just like in the prior experiment. And then we gathered their RNA expression per cell. So we got 50,000 cells, 30,000 data points per cell. And then we have all this in sort of a database, and we asked the computer to just group what the computer thinks based on the gene expression are similar cells. And this is what comes up. Immune cells cluster. So this is called unsupervised clustering. And the size is not representative of how many cells they are, because you could have 10,000 cells all on top of each other in the macrophage pool, for instance. So the size more indicates, the variability among that cell type in gene expression. So anyway, so there's a lot of different cell types here, and we tend to in immuno-oncology, at least right now, we tend to focus on macrophages and T cells. And for instance, you can see there's a large variety of different T cells within the mouse tumors. So, we can drill down on any of these cell types to learn more about it. So, we had known for years something very simple, which was that macrophages, and again, this is mouse, but human looks pretty much the same. Everything we do in the mouse, we then do in humans. Okay, so we're not just sort of a mouse lab. And we're doing it freshly, like we take specimens from the OR and we directly analyze them for hours. So TAMS is macrophages, tumor-associated macrophages. This is the percent of immune cells. CD45 means immune cells. So macrophages are the most frequent cell type, making up about 30 to 40% of mouse and human tumors. T cells come next, B cells, and then there's a variety of other cells. So this is all just flow cytometry, where we can look at all these macrophages and determine what at least they're expressing on their cell surface. Here's a stain, and this is what a normal mouse tumor looks like. But here we're using a brown stain to indicate the macrophages. So you can see there's macrophages everywhere. They're diffusely located throughout the tumor. And this is not that unusual for tumors. So the macrophages are like the glue holding tumors together, but they're doing much more than that. So you heard in the prior overview that there's sort of these different buckets of macrophages. And at least this is what was thought to be true up until a couple of years ago. And so the M1 macrophages are macrophages that produce a lot of, let's say, anti-tumoral cytokines, whereas the M2 macrophages are, giving off these proteins called cytokines that can actually downregulate the immune system. So obviously, as someone who treats cancer, we want to try to get more of these M1 macrophages because they're more likely to kill the tumor directly, the tumor cells, and to give off proteins that activate other immune cells. Well, it turns out that this whole paradigm of how we understood macrophages is wrong. Okay, and I list here M1 phenotype, M2 phenotype, but it's more just to show you that it's not true. Now we're zooming in just on the macrophages. This is the single cell gene expression. So this is, I don't know, it's probably 15,000 cells or so, maybe a little more. Each dot represents an immune cell. And in this case, I'm just showing you the macrophages. And again, this is a 2D representation, but the computer in 3 dimensions will place these cells based on similarity or dissimilarity of their gene expression. So in 2 dimensions, this is what it looks like. And you could kind of say, well, okay, it looks like there's a cluster of cells over here. There's a cluster of cells sort of in this lower part of the green, and then there's clearly a cluster here. And while the red cells do have a couple of genes that are considered M1 in the classical sense, they also express plenty of other genes that would be considered M2. And so the M1, M2 story is almost eliminated, even though it was something that was believed for probably, 25, 30 years, something like that. So this new technology has given us a lot of insight. Now, what we really want to do is understand, which cells are the most important to fight the tumor and how we could augment those cells, like cause them to divide or get them to be more activated. And I just throw this in here to try to give you, you want to hear some sort of therapies that we're interested in. And this is one of them. CD40 is something expressed on certain immune cells, particularly macrophages, but also T cells. And this is an activating antibody. So when this antibody binds to CD40 on these immune cells, it actually activates those immune cells even more. And this is sort of a typical mouse experiment that we do. You don't need to know what this is. It's a control. This is just giving the antibody alone. We see not really much effect in terms of tumor weight after 2 weeks. If we give imatinib, the tumor decreases by about a half, maybe a little more. And we've done this a lot over the years. That's pretty standard. And then if we give both imatinib plus this activating antibody, well, you might say that's not a huge difference, but it's only 2 weeks of therapy. But what you notice is under the microscope, the difference is actually much, much larger. So this is our standard staining here called H&E. And here's what the normal tumor looks like. And then here you can see the purple is sort of largely tumor cells. There's much fewer tumor cells here. Trichrome is a stain that stains like scar tissue. So there's much more scar tissue. And Ki67 is cells dividing, stained brown. And you can see there's almost no cells staining brown. So this is something that we're very interested in bringing to humans. Now, when we look at humans, they're obviously not mice. The mice all have the same genetic background. So they're very consistent in what's inside the tumors. When we look in humans, well, this is 75 humans where we took their tumor directly from the OR to the flow cytometer, or we did, in this case, we did our RNA expression of the bulk tumor, and then we looked at this marker of macrophages. And you can see there's a wide variety of macrophage sort of content, if you will, in the human tumors. And, that raises all kinds of questions like, okay, why? Is it the location? Is it treatment status? How could we get more macrophages into the tumors that have very few? So, you know, this is something that we're working on. In the prior lecture, you know, this sort of story was put up there, and this is human data looking at the entire bulk tumor, not the individual cells. And what we found was pretty dramatic. It turns out that the PDGFR mutant tumors have a much higher immune response than the KIT mutant tumors. And these are just a variety. So each column is a patient. And so there's 37 and 24 patients' tumors. Each row is a certain immune gene. And red means high expression, blue means low expression. And, you know, we wrote a paper, the reference is here. We think this is related to how the mutant gene, the mutant protein, PDGFR or KIT, is being handled by the immune system. And it actually correlates with some things we know about KIT and PDGFR tumors. You know, PDGFR mutant GIST often are, not always, are less aggressive, more indolent growing, less likely to develop metastatic disease. These are not absolutes, of course, but this is, you know, on par looking at one group versus the other. So this fits with a lot of what we see clinically. Well, you saw earlier, you know, all the different immune cell types, and I'm just listing a variety of the cell types that we've worked on in the laboratory, and it's a very complicated system. So the tumor cells have KIT signaling, right? That causes them to make this protein, CSF1, that actually attracts macrophages. And some of those macrophages are good macrophages. Some of them are the bad ones in terms of killing the tumor. These macrophages, a lot of them express CD40, and that's why we want to go after that as a target. The macrophages make this other protein, and it signals this other rare type of T cell, which then makes another protein. And we've proven all this in probably 10 different papers. This is a summary. And this is a key cell. Dendritic cells are not very common within tumors, you know, 1% or less. But they're the ones that are responsible for activating what was called the army of T cells, the CD8 T cells. And these dendritic cells present portions of proteins that then turn on T cells. And remember, the tumor is making IDO, which inhibits the T cells. And I didn't talk about the PD-1, PD-L1 system, but the tumor cells expressing PD-L1, which turns off, again, the T cells. So yeah, I'm happy to answer any questions later. I think we're going to move into a more clinical trial discussion to give some of this some more context. But these are the people in my laboratory that have helped with some of this work and our grant funding. Thanks very much.
Great presentation, and Ron, and you have a great ability to explain it in a way that us mere clinicians can and non-immunologists can understand. So thank you. And it's with great pleasure that I introduce Dr. Arun Singh, and Arun is from UCLA Johnson Comprehensive Cancer Center, and has really been pioneering some exciting immunotherapeutic approaches for our GIST patients. Thank you, Arun.
Thanks, John, and thanks for the background from Junaid and Ron. I think that'll be very helpful in trying to understand the clinical trial and some of the science behind the clinical trial that we did at UCLA. The title of my talk is Stimulating the Correct Immune Response in GIST. As you've seen from Doctors Arshad and De Matteo, the immune system is rather complex. And as you can imagine, when you give medications to perturb the immune system, we hope, and we're always thinking positive, that we're going to lead to the correct immune response that's going to eradicate the tumor. But we also have to be mindful that that we can sometimes lead to turning on the immune system in the wrong direction and leading to deleterious effects. And so I'm going to try to point out why the things we're doing are really trying to turn on the correct immune response. If I'm looking in the opposite direction, sometimes it's because I'm using two computers for technologic issues today. At the base of the problem in GIST is that after imatinib, you have a declining amount of benefit with the second and third line drugs. The fourth line drug actually looked pretty good, which is ripretinib. So the slide's a little dated at this point. Our current conceptualization of GIST is rather simple, and the work of Dr. De Matteo and others is really trying to lead us to have a broader view of what GIST actually looks like. So our current concept is very simple. It's this: growth factors bind to proteins on the cell surface called tyrosine kinase receptors, of which PDGFR and KIT are tyrosine kinase receptors, and lead to normal growth. That's a normal physiologic process. However, the mutations in KIT, if you look in the middle panel, the little X is just the cartoon representation of a mutation, shows that turns on the protein all the time. It's like a switch that's constantly on, and it leads to unrestrained growth. The drugs that we use, like Gleevec, Sutent, regorafenib, et cetera, block these mutant tyrosine kinases and shuts down this growth cycle. Oh, there we go. Sorry, too late in my cartoon. At the end of the day, all the drugs that we currently clinically have access to and we all use for GIST patients more or less do the same things, is that they inhibit these tyrosine kinases more or less intracellularly. And that's the general framework in which we are operating right now. A lot of the work we're doing with immunotherapy is based on expanding this framework and trying to add some additional drugs to our armamentarium right now. This cartoon is meant to show what a GIST tumor deposit should look like. The blue lining is the entire tumor, and each individual green rectangle is an individual cell. But it's not a homogeneous population as the cartoon shows. It's actually much more complicated and probably looks something like this cartoon, where not only don't read everything, but really what it's meant to show is that there are tumor cells. But as Dr. Arshad and De Matteo pointed out, there are macrophages, there are T cells, there are B cells in there, and there's a crosstalk. There's a dynamic crosstalk that's going on. One of our limitations is when we study this, we're usually getting little snapshots in time. It's not like we have what happens from hour 1 to hour 12 to 2 days or 3 days later. A lot of the snapshots we have of the immune system are one moment in time. So there are some limitations to what we can do in the lab to study GIST, and that's why things like Dr. De Matteo's mouse model are actually very are live systems for which we can study this disease. So there are some important things we know about these immune infiltrates. These studies that other groups have done tells us that the presence of the immune cells and the specific type of immune cell has a clinical meaning, in terms of it seems that if you have more or less of certain types of immune cells, patients seem to do better or worse. As Dr. De Matteo alluded to, imatinib is doing other things in GIST besides targeting the tyrosine kinase, as I showed in the original cartoon. It has interactions with the supporting immune cells in the tumor microenvironment. It can inhibit this immunosuppressive protein called IDO, but it also has other groups have pointed out it has activity against that might change the population of the immunosuppressive macrophages in the tumor. So there's a lot that imatinib is doing besides just targeting the GIST cancer cell itself. This is, Dr. De Matteo has gone over some of this already, so I'm not going to go over this again. And so a short summary of what I've presented so far is there are many immune cells in GIST. They're doing something both positive and negative. And as I mentioned just now, Gleevec is not only inhibiting KIT, but it has a dynamic interaction with the other supporting immune cells. Before I go into the study that we've done, what other clinical evidence has been presented before about an active immune state in GIST, and what have we done in the clinic? This is a study that was done at MD Anderson. You see Dr. Trent's name on there when he was a faculty member at MD Anderson. The short story for this is, besides giving patients with advanced GIST imatinib, the MD Anderson group gave them interferon as well. Interferon, in many ways, is an immune stimulant. When you have a viral infection and when you feel weak and you have a low-grade fever or high-grade fever, and all the bad feelings you have, a lot of that is mediated by interferon, which helps to stimulate immune cells, or it can be from the activated immune cells rather. And in this study, what it shows is that when you that the immune cells in the tumor microenvironment actually become activated when imatinib was combined with interferon. These actually, I'm not going to go through all the panels here, but really it shows that this combination seems to really be eradicating the cancer cells in the GIST cancer cells. That's a nice way of saying it. Another study is a small study from Sweden. And in this study, they gave these cells called dendritic cells to GIST patients. And what are dendritic cells? Dendritic cells are some of the supporting cells of the tumor microenvironment. They actually are, you can think of them as a coach. They kind of coach the T cells. They also coach the macrophages too. And really to try to get them to give the right and the correct type of immune response. In this cartoon, you can see that if you start at 1, if you inject this thing called ilixadencel, I think that's the right pronunciation, which is a modified dendritic cell. It's supposed to lead to the recruitment of immune cells that become activated. They present or coach the T cells or help to bring them in to where the cancer cells are to elicit an immune response. And this study was it was a very small study, only 6 patients, but I think there were some interesting things that we can learn from this. One, we saw that, so what was this? This study is for patients who were on Sutent or regorafenib who were progressing on those drugs. So these are people who are progressing on these TKIs, and into the GIST tumors, this modified dendritic cell was injected directly into the tumor. Didn't work for everyone, but you can see here in the first panel A, you see 2 patients who actually their tumors actually shrunk a little bit. These are patients 6 and 7 in the bottom panel here. And you can see that this patient's tumor shrunk about 1.3 centimeters. And the second patient shrunk about 7 centimeters. And these are the 2 patients who actually continued for almost a year and beyond a year on treatment. And so this is interesting because these are people who are progressing on Sutent and regorafenib, that once you added on this dendritic cell, their disease actually stabilized and shrunk a little bit. And it didn't work for everyone. You can see in panel A that the 4 in red, that their tumors grew. And a more descriptive more descriptive graph of the change in tumor size you can see in panel C, where the blue lines just really stabilized out over time, whereas the other 4 patients grew. So again, this comes back to this idea about, well, for some patients, you know, the right type of immune modulation will lead to a good clinical benefit, but also the wrong type will just not help very much. And the other, oh yes, this is another study for another type of immune therapy. And this is what is called PD-1 therapy, or Keytruda or pembrolizumab. This was done in France, but in this study they also gave cyclophosphamide, which is an immunosuppressant. This is a long story, but based on preclinical work, they thought that this was a reasonable way to proceed. They only saw one, really in this study, 1 of 10 GIST patients, only 1 received clinical benefit. And so that compared to our study, this was, this is a small study, but it seemed that not as many patients as we would imagine should have gotten some clinical benefit. So again, the right mixture is what's important about these small studies that we're trying to piece together about learning to manipulate the right type of immune response in GIST patients. So again, bringing the right type of immune cells into the tumor microenvironment could be beneficial, and it seems to help destroy tumor cells, as you saw in the interferon study, as well as the dendritic cells. But giving the wrong thing like cyclophosphamide could be deleterious. Certainly we're seeing we can rev up the immune system in GIST, but we want to be careful not to do the opposite. So getting to our study, this is conceptualized based on the preclinical work that Dr. De Matteo and many other people have done, but also we had some exceptional responders. This is a patient who went on hospice and came off hospice after going on nivolumab, who just had this tremendous response to nivolumab, which is kind of like a cousin medicine to Keytruda or pembrolizumab. And this is the name of the study. Actually, just today, last night, it was actually published in the proper form on Clinical Cancer Research, if you want to take a look. So these drugs really, I'm not going to go, I've been through a lot of cartoons already. I'm probably running out of time, but really they stimulate the immune system in both the tumor microenvironment and as well as the lymph nodes. So how do we measure benefit, and what are good ways of telling who's going to benefit from immunotherapy or not? And one way is that the longer you're on immunotherapy, that's a good thing. That means your disease is not progressing and you're doing well. And the patients in the nivolumab arm, 5 out of 19 patients were on treatment for more than 6 months. And that's it's about 26% of patients. There are some other patients that are on for about 4 months. In oncology, we use markers like how long can you be on treatment for 4 months or 6 months as good measures of people receiving clinical benefit. And mind you, all of the patients who came on this trial were failing all the standard treatments that are available and had progressive disease. So these are not people whose disease were stable coming on to the treatment, to the trial. And you can see that there's a patient that was on for nearly 2 years, the top patient as well. And the second arm, patients received 2 types of immunotherapy, nivolumab and ipilimumab, which goes by a name called Yervoy. And you can see that there's some patients who were on for 2 years. One is, I guess, close to getting probably close to 3 years at this point, and she still has not recurred. Another patient over 2 years, and another one for about 8 months. A couple other patients were just under the cutoff for 6-month benefit. So again, even with both arms, regardless of which one we gave, there's some patients who received a lot of benefit. But these are small numbers. These are not large trials. You know, this trial from conceptualization to now, it was since 2015. It's been 6 years from the conceptualization of this trial to the readout that we're getting now. So as Dr. Trent mentioned, you know, things in the clinic move slow compared to the lab. But one of the real goals of this trial was to really establish what a baseline is for how well do these new drugs perform for patients with GIST, because we see these drugs are approved for several different types of malignancies already, and across the board, they're active in about 20% of all malignancies, and so of all patients with several malignancies. So again, the goal, and again in GIST, we're seeing that at least, here, I'll go over the numbers with you in just one second about how many patients and how we stratify who's receiving benefit. We don't get a ton of tumor shrinkage, but there are some patients that do get shrinkage. And it's important to try to study those patients and understand more why they're getting shrinkage. This is the patient who was on going to be probably 3 years soon. On the left, you see about a 5 by 4 centimeter recurrent lesion that you just cannot see. At least this is only about 2 years later. You could not see exact any residual tumor at that point. This is a PET scan. Patients and some other doctors have asked for PET scans, what the PET scan changes look like for these patients. And you see quite a large tumor on the left on panel A, and panel B is the corroborating PET scan to this lesion that you had a little bit of shrinkage, not a ton of shrinkage after some treatment, but you can see that the rim of the tumor is not as warm as before. The yellow areas have gone away. And so showing that this patient had tumor shrinkage. The patient already had been having necrosis in the center of the tumor, but the amount of tumor volume did shrink. And so these are important clinical correlates as to really for clinicians to understand, hey, there's a group of people that can get shrinkage and can get good responses by how we measure things in the clinic. Again, overall, we think that about more than 50% of the patients in nivolumab got some degree of clinical benefit. And in the nivolumab and ipilimumab arm, we saw maybe about 30% or so of patients got clinical benefit. This is a small study. We have to be modest about that, and that's the way we operate in the clinic. We don't want to make too much of this, but this is 36 patients. There's one patient that was not evaluable. It's a small study by clinical standards, but for rare diseases, this is how we operate. We operate with smaller numbers, and we're trying to understand this new therapy to change the paradigm of our treatment options for GIST. These are some of the patients who had long-term benefit. And a lot of people have asked me, why are some patients responding and others? And we're trying to understand. People have had, it didn't really matter which therapy you came off of. That's something people ask. You can see here, there's a whole variety of mutational types, exon 9s, exon 11s, double mutants, and a wild type patient as well also received benefit. One thing I did see is almost most of these patients had their disease start in the small intestine. The one that quote the ovary and mesentery, we don't really know where that patient's disease started. One thing that was pointed out is that, there's some of the patients who were very sensitive to the tyrosine kinase inhibitors. I think Dr. Trent will probably be interested in is that if you had a lot of sensitivity to tyrosine kinase inhibitors, that means you got kind of what we would describe as an allergic reaction to some of the tyrosine kinase inhibitors. You might have a better chance of responding to immunotherapy. And certainly some of the patients who had really long-term benefit did seem to have that type of body makeup, or phenotype is the term that we use. This is, we've tried to study what happens with the types of cells we're seeing with change. This is a patient on the left. You see that before they received treatment with the immunotherapy, the green is the GIST itself. The red is the T cells or the immune cells. And on the right, you see a biopsy after treatment. And all of a sudden, what's the difference there? What's all the red in the middle of the tumor now versus not being very present at the beginning? So we think that some of the patients who are receiving benefit is that because when you got the immunotherapy, it brought the cells into the area, the T cells, those are the magic cells to get the active T cells into the tumor microenvironment. But we need to do more work. And one of the surgeons, Joe Crompton, who came from Sloan Kettering, is going to work on this as well to try to understand this a little bit better. I did mention that that we did have a wild type patient who received benefit, and this is also, I want to let other people know that Dr. Pollack did publish a case of another wild type patient who did receive benefit, and you can see some tumor regression, tumor shrinkage in their patient with wild type GIST who was on nivolumab. That's something we should know. There are other types of immunocombinations going on. I want to thank the sarcoma group and all of the study coordinators and people who helped the study happen. Conflicts of interest. There you go. That's it. That's it for me today.
Thank you, Arun, for sharing the great work that you've done in helping develop immunotherapy for GIST patients. At this time, I'd like to open the panel for discussion. And there are some questions in the chat already. And the first question is about immunotherapy, specifically for exon 11, 557, 558 deletions. I don't know what, maybe Ron has some thoughts as to whether or not the immunogenicity or the potential for immunotherapy would change based on site of mutation in the KIT gene?
Yeah, I don't know the answer to that. I mean, it's certainly something of great, interest to us. We just need to analyze a lot of patients to be able to say that there's differences, related to where exactly the mutation is. I mean, we do know in the mouse models that different mutations lead to different sized tumors and different activity. So I would imagine that that carries over into different effects on the immune response, but we just don't know that yet in humans.
Great. So there's another question from an anonymous attendee. Any thoughts on precise monoclonal antibodies of PD-L1 inhibitors and TILs from other indications? How many T cell receptors may be required for therapeutic efficacy? There's lots of data in other tumors on single cell sequencing. What are your thoughts, Ron?
I mean, certainly we use, we know that there's other tumors that have, been investigated by many people. So breast cancer, renal cell cancer, a few others. So, we use that information to try to help put the GIST findings into context. With regard to the antibodies, well, the companies have put out mostly an immune checkpoint blockade, but which is the IPI and the Nivo that you saw Dr. Singh present. I mean, there's a whole list of other inhibitory receptors that exist in the immune system, and I think you're going to start seeing a lot of these other sort of blockade type strategies. And then there's a bunch of activators like the CD40 that I showed. So
What do you think about T cell receptor density? I think part of the question had to do with TCR density. Is that something you evaluate in your flow cytometry?
No, we haven't. Like T cell receptor density on a given T cell.
Yeah.
No, I mean, the problem is we don't even know what exactly the antigens are that the T cells are responding to. So we've begun doing what's called TCR profiling, where we can, because for the audience, each T cell is only looking for one specific target. And that target is one little piece called a peptide of a protein. So we have all these T cells in our body, and when a T cell gets engaged in the right way, it starts dividing to make more T cells specific for that one little piece of protein. So we don't know yet what the antigens, like what the T cells within the tumor are reacting to. But I didn't go into that technology. There is some good technology, and we've started doing it, and we can compare the receptors on the T cells, in other words, what that T cell is looking for, with the blood and the nearby normal tissue and all that. But for most human tumors, we don't know what the T cells are reacting to yet, but that's a huge area of investigation.
Well, that's fascinating. I had a question for Arun. Do you, how does imatinib side effect of lowering white blood cell counts, and how does that play into the potentially using immunotherapy treatments? Do you see that as a barrier from your experience and on your trial? Was there, do you think, was that a concern?
Yeah, so certainly with imatinib, we do see a drop in the total white blood cell counts in many patients. But not only and actually, if you look closer, you can see a variety of things. You can see drops in things called neutrophils, and you can see drops in things called lymphocytes. The lymphocytes are the CD8 and CD4 cells. So in some ways, yes, there's a relative immunosuppression that is happening. And the question is that clinically meaningful? And what does that lead to? First of all, in general, for the last decade of treating GIST, with not just with imatinib, but you also can see this with some of the other drugs as well, we don't really see clinically significant infections in GIST patients. I don't know if you all can add to that as well, but generally, despite this relative immunosuppression, there's not a clinically significant set of infections that happen.
Great. That's been my experience too as well. I wonder, Junaid, if you're aware of any mRNA technology, messenger RNA technology being studied as a potential to deliver immunotherapy to GIST patients. You're muted.
Always forget that you have to unmute first. So it certainly seems like a very good concept. And if you, like I said before in my presentation, that it can be in the form of vaccines. And there have been some work prior on vaccines in the form of messenger RNAs for other sarcoma types. But I'm not sure if there's anything going on, and specifically in the field of GIST related to messenger RNA. And part of the reason being also we have to determine the effective targets for these messenger RNA treatments too. So currently I'm not aware of anything that's going on.
Now there was another clinical question. Should those on adjuvant therapy for high-risk patients at this point continue on imatinib indefinitely until immunotherapy advances come to fruition? I think that's an important question. There are certainly a subset of patients with high-risk GIST for whom we do leave on imatinib indefinitely, specifically if there's tumor rupture and some other features. But if it's a patient who clinically we would discontinue imatinib after the 3 years of adjuvant therapy, I think that's still a reasonable approach. I personally don't think we should continue therapy that we think is unnecessary until immunotherapy comes to fruition. Also, the reality is that based on the French discontinuation data, when imatinib is discontinued and a patient develops progression, the probability of shrinking the tumor again well exceeds 90%. So I think that I would, I'd certainly, whoever asked the question, I would consult with your treating physician and have this discussion with them. but I don't think the potential of immunotherapy would change my clinical prescribing pattern at this point in time. I don't know if anyone else has any thoughts on that topic.
Absolutely, and yeah, I completely agree with John. But I would also say, this is a very individualized setting, and there are some centers that don't take people off adjuvant imatinib as well. So it's really a discussion you need to talk about the risks and with how well you're tolerating the Gleevec, which is really the limiting reagent for patients who are going to come off at this point.
Well, we have 5 minutes left, so I'd like to pick the brains of each of you. We can start with Ron, and the question from the audience is, what do you think the field is going to look like 3 years from now?
Yeah, that's a great question. I need a crystal ball. I would like to think, the tyrosine kinase inhibitors have been a great story, right? The incremental advantages, are probably diminishing to develop additional ones. That's, I think it's clear to most people that we need some sort of combination therapy. And that other combination, it's not going to be a replacement of a tyrosine kinase inhibitor, but what that combination is, immunotherapy is one option. There's other drugs that have novel mechanisms of action. I'd like to think immunotherapy, at least for a subset of patients who we can identify, could be very useful. And you've seen in some of these case reports and Dr. Singh's trial and other, there does seem to be a group of patients that benefits greatly. And I'd like to see personalized immunotherapy at some point with tyrosine kinase inhibitor. I mean, that might not be 3 years, but hopefully in the 5 to 10 year range.
What did you think? You have any comments, Arun?
Yeah, so certainly I think with this data, we saw that there's a subset of patients who really receive benefit. And really, we're going to really look into those patients as to why they're receiving benefit, and try to pick out those patients that we can start combining with TKIs. I think that's certainly the next step. The hard part is now that there are 5 TKIs, and so which one do you want to give, and in which line? First line, second, third, or fourth line? Doing it later on is fraught with difficulty because the patients are not doing as well, and it's hard to get on a trial because you have to wait 2 weeks to get on a trial, which a lot of patients can't afford. So the decision has to be, we really have to cherry pick who's going to benefit to really stack the deck and make this a really viable therapy, in my opinion.
Great comments and great thoughts about the future. Junaid, any closing comments?
Yeah, so to answer your question, Dr. Trent, so definitely, the future does hold the role of immunotherapy. It's just we have to determine in what set of patients these treatments work the best. So, and essentially, there is a role of discovering new targets as well, and new treatment combinations with better side effect profiles too.
Yeah, I wholeheartedly agree with each and every one of the panelists. I think we have exciting combinations in the future. I would like to see immunotherapy combined with KIT inhibition, and possibly including even some other mechanisms such as induction of apoptosis, metabolic pathways. We're still discovering a lot about GIST and hopefully more effective therapies. And I know each and everybody on this panel is working hard so that we can move the line of scrimmage forward in the next 3 years. And so I'd like to end by thanking the panelists for taking time out of their busy day to participate in this important educational event with the LifeRaft group. And I'd like to thank the LifeRaft group for sponsoring this event and doing all of the logistics and arrangements so that it was successful. And ultimately thank all of the attendees at this conference. And thank you for taking time out of your day and spending time with us discussing the